Thank you so much for this invitation. First of all, we will introduce ourselves. I am Professor Marlies Eekhoff from Amsterdam. I'm leading the Rare Bone Disease Center, where we aim to develop and help to develop drugs for rare bone diseases. Of course, we collaborate with many pharmaceutical companies, but we also do other trials, and that is STOPFOP we will speak about today. I pass over to Vincent.
So my name is Vincent Verheijen. I'm a physician also at the Amsterdam UMC. I'm here also talking about the STOPFOP trial in which I am the lead sub-investigator under the PI Marlies Eekhoff. Pass on to Chris.
Hi, so first of all, I'm going to say I'm probably the only person here who's going to have a script. If I didn't have this, I would forget my name. So on that note, my name is Chris Bedford-Gay. I am dad to Oliver. Oliver is my eldest child of three boys, who in 2009, at the age of 1, was diagnosed with FOP. Oops. Those 3 letters that combined to make one of the rarest, most disabling genetic conditions known, 3 letters that changed our lives. FOP is an ultra-rare genetic condition, a condition that allows us to actually say our son is 1 in a million. Post-diagnosis, we set up FOP Friends, a UK charity, and we were lucky enough to have a UK-based research team at the University of Oxford. We began raising funds to keep that team going. We've been doing that now for just over 12 years, with a little bit of help from our friends at FOP France also. We also support our families, trying to make life with FOP the best that it can be. But what is FOP, you might be asking? For FOP, or fibrodysplasia ossificans progressiva, is a variable but irreversible genetic condition where muscles, tendons, and ligaments and connective tissue progressively turns to bone. It is also trauma-induced, which means that there is no way to undo FOP's progression. Children are usually impacted within their first decade of life, severely restricted into their teens, completely immobile by their 30s, and with a reduced life expectancy. Progression, the unwanted bone growth, is usually preceded by an inflammatory response, tumor-like swellings, a signal that FOP is about to do its worst. But diagnosis is easy, with a little knowledge. So you forgive me for a moment to explain that in 97% of cases of FOP, from birth, they have turned in or malformed big toes. That should raise the question, could it be FOP? Combine those toes with tumor-like swellings, then it's almost certainly FOP, and a genetic test exists to check. We can see that there is an urgent unmet need, therefore, for a treatment. FOP is severely disabling, life-limiting chronic condition. Most with FOP are restricted within their first decade of life. Immobile by the teens, I'm repeating myself slightly, reinforce the point. Bed or wheelchair bound, not long beyond that. Lungs restricted, jaws locked, impacted by scoliosis. No treatment within Europe, and no globally available treatment. As an ultra-rare condition, there's also no guarantee that industry alone will get us to that treatment. Now, this is where it gets a bit interesting. Our relationship and support of Professor Alex Bullock's FOP team at the University of Oxford and our trust in that team allowed them the time and resources to take basic research findings and screen a library of existing but unused drugs as inhibitors for FOP. Our team were reimagining medicines. Unfortunately for us, Dr. Eleanor Williams found a likely drug candidate, saracatinib, a drug I learned today that is being repositioned, not repurposed. A drug candidate that looked like it could actually stop FOP.
Saracatinib, what is it? It's a protein kinase inhibitor. It's an experimental drug being developed by AstraZeneca, original for cancer. No one ever considered it to be that it could ever play a role in FOP. In FOP, it's important to understand, a very short slide, that the mutation, the pathogenic variant, involves the receptor. And that the main receptor is the BMP1, which plays a very important role in the cell for the development into normal muscle. But in FOP, it is mutated, and instead of muscle, bone is being formed. And here you can see it below that it is a part of the muscle, of the part of the receptor, the ALK2, that is really affected. How could we treat FOP? There are several options, but one of the options is that you directly block the affected region of the receptor. Well, Alex Bullock, with help of the patient organization, looked whether a lot of compounds, how they bind to the affected region. And they found that saracatinib was by far the most strong bound to the receptor. And they also showed that it could block actually the affected receptor, and also in a mouse model, FOP mouse model, they showed then compared to placebo that it could prevent bone formation. So it was very, very promising. And that's why we started the FOP trial. We knew each other from the European FOP consortium. And on the left side, you see executive sites, Amsterdam, but also Richard King, Professor Richard King from London, and Clemens Stokklausner from Garmisch-Partenkirchen. And on the right side, of course, they are part of the team, Alex Bullock and Paul Lewis preclinical team, and in collaboration with AstraZeneca. And the trial is called saracatinib trial to prevent FOP, STOPFOP. Luckily, we got granted, got money, 1 million from the EU IMI, but now it is called Innovative Health Initiative. Sorry, thank you. And we did it with we set up the trial with all the partners, but so importantly, also with all the FOP patient organization from Europe, UK, and from the United States, IVOPA. We also, I have to say, also got from them not only support in thinking along about the trial, but also financially. And also we now got money from LifeArc, and we are very, very, very happy for that. University-driven trial, halfway, then I have 2 minutes still, I think. But of course, it has been told a lot. You're free to set up the trial yourself with direct involvement of the patient's voice, direct collaboration, support, input. You are doing the trial from beginning to the end. And I think without passion or motivation, you would never be able to do such a lot of work. And always the money is not enough, so you're doing a lot in your free time. We feel very independent. We are very happy to collaborate with the patients organization. And they give us a lot of support, financial and in the trial itself. We come up to it later. Repurposing, I think I should change it now. But why is it so good that we could start with saracatinib? Because it has such a high profile. It has been tested in more than 700 patients. So we immediately could go on in the trial. And you will tell more. No, the next one is yours. So I give over to you why the safety profile could help us to start the trial in phase 2. Yeah.
Thank you, Marlies. So up to me to tell you about the current status of the trial, the design, and where we hope to be in a year. Currently, as Chris said, there is no treatment for this horrifying disease. It's also referred to as Stoneman syndrome because patients literally petrify. They fuse in their body, joints fuse, including their jaws. And they're usually wheelchair bound at 20 to 30 years old. There is nothing we can do for them so far besides corticosteroids. If the inflammation that results in ossifying immobilities involves larger joints, then we sometimes give them corticosteroids, but there's nothing else that we can do now. Luckily, there's 4 trials going on in the world. 3 that are sponsor initiated and 1 that is investigator initiated, which is ours, the STOPFOP trial. It's a small trial. We only have a few patients. We had limited means. We are considered the underdog, but I think we all know what another drug can do. The good thing about what we did is that the saracatinib drug or the compound has been developed 25 years ago by AstraZeneca, and it's been tested in more than 30 trials so far, phase 1 and phase 2, which is why we could go into phase 2 immediately and screen whether this was safe and both efficacious in our very delicate population. So we're doing a phase 2 trial. The aim is to investigate the effectiveness or the efficacy of this compound, of course, but moreover also the safety of the compound. It's a very delicate population. Patients have restrictive lung disease. They can often not eat because their jaws are ankylosed. And so we want to be sure that this compound is safe, doesn't result in any more inflammations or what we call flare-ups and more ossifications. And we're also, of course, looking to see whether it's efficacious or not. But I think we will need to go on to phase 3 before we can get market approval. But both efficacy and safety will be tested in our trial. We're doing that in 3 locations. So the Amsterdam site here at Amsterdam University Medical Center is open. We've been seeing patients there for 6 years now. The last 4 years, we've also been including patients in Garmisch-Partenkirchen in Germany. And we were hoping to open another site in the UK, but then Brexit occurred, and obviously the COVID pandemic messed things up. So we have 2 sites opening that are open now. We've closed inclusions where we're almost to the point where we're analyzing our data, but we have had patients coming in from different countries in Europe, which I'll show later. The trial itself, it's a 6-month RCT, 1-to-1 randomization for either placebo or saracatinib, so the drug, followed by a 12-month open label extension, which was very important for the patients. They are all very limited in their movement. They all know they have a progressive disease, so they all wanted to have at least a chance of getting the drug. So they all wanted to participate as long as there was an open label extension to the trial. So 6 months RCT, then followed by 1 year open label. And then we amended the protocol that as long as the last patient has not had his or her last visit, patients were allowed to continue the drug until the last patient last visit under the Helsinki Agreement, which says that you can continue medication or the drug until negative data or alternative treatments are available. Here you can see a map of Europe. In orange are the countries that we've been able to include patients through the Amsterdam site in, and then in Germany, we've also been including patients from both Austria and Germany. We have 18 patients included, 17 who are still active, and all the subjects who have finished the formal trial, so the 6 months open label, the 6 months RCT plus the 1 year open label, have all opted to continue the drug after the trial. And I think that's what tests for, at least subjectively, the power of this compound. There have been previous trials in this disease with over 60% of patients opting to get out of the study because of side effects or no apparent effect. But our patients have been desperate to continue on this drug. And that's what I think shows how important it is that we finish this trial, albeit small, albeit there's only a few patients, albeit us being the underdog. But I think it's gold that we have in our hands. So we hope to have the first analysis within the next few months, of which we will be presenting hopefully at the ASBMR this year in America, the largest society for bone mineralization research. And we'll see what happens afterwards. We've been doing this with a very limited budget, with so little help, but I'm very proud of what Professor Akov and all the patient associations have been able to do for this population. I think we should conclude with why our collaboration was so important. I'm not sure whether you can attribute to this, Chris. I'll pass back on to you.
Thank you. Yeah, so I wrote down some notes that essentially, why is it so important? It's possible for us patient organizations to actually have a seat at the table. We are a valued partner in this relationship. It allows us not only to contribute, to engage in, but it also allows us to help our community in accessing trial opportunities they might not have already had. It's allowed us as a patient organization to really play a part, and actually the hope that it gives us and therefore our community is almost immeasurable. We are empowered, and we know then that we can actually make a difference. We know we're being listened to. We're not just talking into a black box, which can quite often happen. You know, we'll give an opinion and then a trial design will come out or something will happen. hang on, we said don't do that, or include this. And so we really get it involved. So STOPFOP in itself has and is an amazing achievement. It's kind of an example of what can be done. It's hard work. It's hard work for us. Exceptionally hard work for this team with the resources and the budget that they've had to do. And it's just a testament to how we are really stronger together. What I should say, I've kind of talked a little bit about us as an organization, FOP Friends, and where we were involved. But actually from the start, there's been a stakeholder group, which is a lot of the national FOP associations from around the world who were brought together. I think almost always in the Netherlands, alongside the Dutch FOP associated meeting, where our opinions were sought, along with all of our scientists, all of our researchers, including AstraZeneca and drug developers. We break out in sessions, we discuss what is important to us, we feedback to everyone, and it gets all taken into account. It all makes a massive difference. We don't really get that anywhere else. We get asked our opinion. We volunteer it because we need to get to our end game, but we really feel included. So it's really made a big difference, and it does empower, it does motivate. And as patient group leaders, quite often you're rolling a rock up the hill, you're trying to walk up the down escalator, that kind of stuff. When you get together in a collaborative environment like this, it makes all the difference. It re-energizes you, it keeps you going. Yeah, that's it.